Welcome back to Anatomica. In these sessions, we will be discussing histology of cartilage. The learning objectives of the sessions are discuss the general features of cartilage, explain the cells, fibers, and ground substance of cartilage, explain and differentiate hyaline, elastic, and white fibrocartilage, explain the degeneration and regeneration of cartilage, describe ossification and calcification of cartilages with age. It is a special form of dense connective tissue present in places where firmness, elasticity, and flexibility are needed. The cartilage present at the tip of the nose or auricle of the ear readily demonstrate that cartilage is sufficiently firm which maintain the shape of the structure but is flexible and not rigid like a bone. Try bending your nose and ear. It can be bent in any way. But once the force is removed, it comes back to normal position. During development, in the intrauterine life, the cartilages present in developing long bones are replaced by bone and are called temporary cartilage. In places where it persists throughout life as cartilage are called as permanent cartilage. Like other connective tissue, it also develops from the mesenchymal cells. Cartilage has its own peculiarities. The cartilage is avascular. Then, how does it get its nutrition? It gets its nutrition and gases from the surrounding tissues with the ground substance providing the medium for diffusion of nutrients and gases. The source of nourishment is the blood vessels present in the perichondrium. The articular cartilage in the joint cavity, which lacks perichondrium, derives its nourishment from synovial fluid that is present in the synovial cavity. Since the cartilage is avascular tissue and it depends on the surrounding tissue for its nourishment, it has poor healing capacity. This also explains why cartilage cannot become very thick, since diffusion of nutrients and gases to the deeper tissue will be insufficient. It also lacks lymphatic vessels and nerve supply. It exhibits tensile strength where it can withstand maximum stress. It is resilient where it is capable to return to an original shape after being pulled, stretched, and pressed. Function depends on the nature, proportion of ground substance, and extracellular matrix. The function is specific. The functions will be discussed along with respective cartilages. And finally, it provides firm structural support for soft tissues being flexible. Coming to the components of cartilage, under the components of cartilage, the whole cartilage is covered by outer connective tissue sheath called perichondrium. It has two layers, outer fibrous layer and inner chondrogenic layer. It consists of cells and extracellular matrix. The cells of cartilage are chondroblasts and chondrocytes. The extracellular matrix is abundant and consists of fibers and ground substance. Let us discover the details of the components one by one. Let us start with details of perichondrium. The picture on the upper left is the photograph of the section of the cartilage. The lower picture is the diagrammatic representation of the cartilage. The whole cartilage is covered by perichondrium. It is a dense connective tissue which is essential for growth and maintenance of cartilage. It is derived from mesenchyme surrounding the developing cartilage. It consists of an outer fibrous layer and an inner chondrogenic or cellular layer. The outer fibrous layer is formed by collagen type 1 fibers. The mesenchymal cells surrounding the cartilage differentiate into fibroblasts. Fibroblasts form collagen fibers and together they form the fibrous layer of perichondrium. Chondrogenic layer or inner cellular layer is made of fibroblasts and chondroblasts. This layer is present in the deeper aspect of perichondrium. Among these, fibroblasts in the inner layer of perichondrium are progenitor cells for chondroblasts that divide and differentiate into chondrocytes. Perichondrium has blood supply, lymphatics and nerves, whereas cartilage does not have these and is avascular. Hence, oxygen and metabolites diffuse from the perichondrium into the cartilage. To start with first component, that is cells, chondroblast is immature small dividing cell. It is oval-shaped cell and is present in the peripheral part of the cartilage. It is oriented parallel to the surface of the cartilage. It is precursors of chondrocytes. It is metabolically active with euchromatic nucleus, prominent or abundant mitochondria, Golgi complex, and endoplasmic reticulum. Chondrocytes are mature cells or resting cells. The shape of these cells varies in different regions of the cartilage. In the peripheral part of the cartilage, they are oval in shape, whereas in the deeper zone, they are somewhat round in shape. They are present in small cavities in matrix called lacunae. They synthesize and secrete extracellular matrix. They have heterochromatic nucleus and organelles become less prominent. We finished perichondrium, we finished cells, and now we are in the third component of cartilage, extracellular matrix. It is made of ground substance and fibers. Ground substance, it is well hydrated. This large component of water provides resiliency to the cartilage. Because of the resilient nature of the cartilage, it can get distorted without any damage and hence it provides flexible support to the surrounding tissues. It also provides the medium through which diffusion of gases and nutrients take place. Ground substance is made of water, as already discussed, then carbohydrates and proteins. The carbohydrates are formed by the glycosaminoglycans, also called as GAGs. They are classified into long and short GAGs. GAGs, which are long mucopolysaccharides, include hyaluronic acid, and short GAGs, which are sulfated, include chondroitin sulfate, heparin sulfate, damatine sulfate, and keratin sulfate. The proteins are formed by glycoproteins and proteoglycans. Glycoproteins have multiple binding sites for cell receptors and for other matrix macromolecules. Example is fibronectin and integrin. Proteoglycans are present in abundance in cartilage and are tightly bound. Proteoglycans are large molecules consisting of a core protein to which numerous sulfated glycosaminoglycans are attached. The core proteins are aggregans. Fibers. Okay, coming to the fibers are present in cartilage are collagen fibers, which are distinct, and elastic fibers. Collagen fibers present in cartilage are type 1 and type 2 fibers. Perichondrium and fibrocartilage have type 1 fibers, and the rest of the cartilage have the type 2 collagen fibers. Elastic fibers are more abundant in elastic cartilage. The details of the fibers have been discussed in the general part of connective tissue. Please go back and watch the video for details. How are these components of cartilage arranged? Imagine ice cream, and within this ice cream, the nuts cut into pieces which are very thin pieces, and choco chips inside a cone. Imagine outside cone as perichondrium, ice cream inside the cone as the ground substance, thin pieces of nuts as fibers, and choco chips as the cells. In a section of cartilage, there is a perichondrium covering the whole cartilage, deep to which is ground substance having water, carbohydrates and proteins. In this ground substance, cells are arranged in groups or rows which ranges from 2 to 8 cells. Around the cell groups is a space called lacunae. In the living, the cells occupy the whole of lacunae. In preparations, due to the shrinkage of the cells, the lacunae can be seen. The fibers are embedded in the ground substance interspersed between the cells. Fibers present are collagen type 1, 2 and elastic fibers. So till now we have discussed in general the components of cartilage. Okay, to recap, we have gone through introduction, components like cells which are chondroblasts and chondrocytes, extracellular matrix which includes fibers and ground substance, and finally the arrangements of these components in general. In the next session, I will be explaining about each type of cartilage in specific.